Yeah, right. I guess we need some guidance on how to interpret it.
Yeah, we can do that. That's not a problem.
Okay, good.
This is the Convergent Science Network podcast.
But you would be interested in doing that?
Yeah.
Okay,
Leading
because
researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
It's Paul Verschure with the Convergent Science Network podcast, and I'm here with my colleague Tony Prescott in the BCBT workshop of 2014, and we were with our speaker Henry Kennedy, who has been talking about the architecture of, in particular, neocortex. And Henry, you sort of you came in not sort of opening the door, you sort of knocked down the door by just saying, look, all the stuff you guys know and love is wrong, like small world networks, you should take with a big grain of salt. Standard ideas about how we think about the connectivity of the human neocortex are maybe incorrect, right? So what's your position exactly towards these standard views on?
Not that the small world network is wrong. It's not. It's simply a proposition based on inadequate data.
Mm-hmm.
So if you look at the complete data of interaerial networks, it just so happens that they're very high density, and at that kind of density, the small world model is not appropriate. It doesn't tell you anything about it. So actually, what we're trying to say is that what does give you a much more meaningful explanation about what the architecture corresponds to is if you take into consideration the distance and the weight, the strength of connections. And this breaks away completely from the small world tradition because you're no longer dealing with a binary network, you're dealing with a directed, weighted network.
Mm-hmm.
That was the point we were trying to make.
Right. So you're saying it's just too crude a view to really help you understand how a cortex is wired up.
It's ignoring too much about what actually specifies the network. If you have a network which has a density of 70%, it's not what is connected to what that will tell you very much. It's how strongly which area is connected to which area. It's the strength of the connections, and it's a wide range of strengths that makes the cortical network so very, very surprising in some ways. There's a big range of strengths of connections. They cross over 5 orders of magnitude, and it's taking those strengths into consideration that you'll have an insight into what is actually the fingerprint, the connectivity profile of a particular area.
Right. So now you've been spending a lot of time doing very detailed studies using tracer injections in understanding the connectivity, in particular of the cat neocortex, and I guess with emphasis on the visual areas, right?
I started off in cat visual cortex many, many years ago, yes. More years than I care to remember, 20 years maybe since I've touched a cat, yeah.
Right. But just to make the point that you're not basing your conclusions on, let's say, a functional description of the system, or in terms of what you might want to call a functional connectivity. You base it really on a detailed study of, let's say, injected cells, right? So it's a very direct measure of connectivity.
Yes.
Okay. So then, given that data set, how do you think of what is the template you have in mind of cortical connectivity? How is it different from what a small world would tell you? How is it really wired up? What are the wiring rules?
Well, there is a wiring rule, and that we felt was that was the backbone of the publications we've been making recently. There is a very simple, very straightforward wiring rule, and that is that there's a minimization of wire. And we're certainly not the first people to find it, but what I think we have been able to put our finger on is a principle which explains that wire minimization, which is such a strong constraining feature of the cortex. So the constraint basically that we've been able to demonstrate is that there's a weight-distance relationship. So there's an exponential fall of strength of connections with distance. And if you take that on board and you generate random networks based on that particular feature, using the space constants, the lambda value that we've observed, you generate networks which, in many ways, reflect the properties of the cortical network that you can look at down the microscope, that you can actually measure.
Just to clarify, we're talking about a primate.
Yes.
Not cat,
Yes.
aren't we?
Yeah. It's non-human primate.
But then, isn't this contradictory what you said earlier about weights? Because in some sense, so the data is roughly telling you that over a distance of, let's say, 60, 70 millimeters, you would have an roughly exponential kind of decay of the probability to connect and of the connection strength.
Yes.
Yeah. But earlier you said the small world view doesn't help you because you have maybe low probability connections, but their strength matters. Their strength actually can tip the balance, if you want, into a significant functional relation or not. But now your data shows that the weight also drops off with distance.
Mm-hmm.
So would that then not undercut your earlier argument against the small world network?
Well, the point about the criticism about the small world network is simply, if you take the density, that is to say, the number of connections that you have between different cortical areas, it turns out to be about 70%. So 70% of the connections that can exist actually do exist. That's a very, very high density. And at that density, you have a small world.
Mm-hmm.
So you don't have to measure the clustering index, the path length, or what have you. Everything is virtually connected to everything else. Every area is 1.5 hops away. Now, what you're referring to now, the fact is that when you look over very long distance, yes, very few areas are connected. And that's interesting because it means that you have a sort of binary specificity over those distances. Presence of a connection in itself is going to be significant in terms of trying to understand the biology. Our point is that globally, overall, it's going to be the strength of connections which matter. But there are exceptions over these long distances. So, for example, if you take the frontal eye field projection to area V4, that connection is actually rather strong. It's stronger than what you would predict. So it's an outlier, and I think that's something which comes out of our investigations, which I think need to be considered in more detail.
You're really doing the analysis two spatial scales here. So you're talking about connectivity between cortical areas.
Yes.
Of how many are we talking there, sort of?
Well, we're working with an atlas of 91 areas in the macaque monkey.
Right. And then at another spatial scale, you're talking about connectivity within an area, which you're saying is 80 to 90% of the connections are within cortical areas.
Yes.
And then spilling out into surrounding areas with the remaining 10 to 20% of connections.
Yeah.
Yeah. So, but I think when you're analyzing the data, are you analyzing it in different ways when you're doing these two analyses, or is
we haven't the connectivity within the cortical area? This is a local connectivity,
right,
if you will. So if you take a point in a cortical area, 80% of the projections to that point come from within 2 millimeters, actually.
Right.
That's the local connectivity. And we're not looking at that in any great detail. Actually, it should be looked at because there's been very little work on that. So you have the canonical model, which is developed by Kevin Martin and Rodney Douglas. That was for area V1 of the cat. It's never been done for other cortical areas. We don't know what that looks like. We're not doing that.
So when you say most of the connections are within an area, they're actually within a very small part of that area.
Absolutely, yeah. Mm-hmm.
Well, I think the range you were talking about in the data you presented today was, like I said earlier, up to 80 millimeters or something, right? Not beyond that.
Well, this is
for the scaling laws that you showed.
Yeah, for the macaque monkey, it's about 7 centimeters is about the size of the brain. Yeah.
Mm-hmm. Okay. Of course.
Mm-hmm.
But what is then so if we now look at these rules of connectivity that you presented, that seems to suggest that indeed, like you said earlier, everything's connected to everything, directly or indirectly. So that would seem a little bit unspecific to talk about, let's say, an architecture that actually has a certain function. And also when you look at the physiological properties of it, it doesn't necessarily look as some sort of uniform structure. It seems much more fractionated in its dynamics. So So how do you match these two? Right. So is there an element of fractionation and specialization that we just have not seen in this data?
Well, I think you were describing it though, weren't you, with the dorsal and ventral stream? There was some fractionation.
Well, no, this is what I want to get to, right? So how do we get, because just at face value of this Markov kind of data, the scaling laws, you could say, okay, well, this looks pretty uniform wherever I go. It's sort of connected in a similar way, similar kinds of weight distributions, similar kind of length distributions. But how does it give you functional specialization of areas and functionally organized forms of dynamics? That's the question then.
Okay, so the short answer to that is the binary specificity is low, as you pointed out. Everything is not connected to everything, but there is at least within say 15 millimeters of an area, there is a very, very high connectivity. So you are virtually approaching 80%, maybe 90%. So it's going to be the strength of connections which are important. So we've looked at the global properties of these networks. So you can make an efficiency measure, which is a sort of conductance where you're treating the strength of connection as an inverse resistance. And when you do that, you look at the local efficiency and global efficiency, and then you can look how the global efficiency, the local efficiency is affected by thresholding, removing the weakest connections until you have just the backbone of very strong connections. And then look how your efficiency changes with removal of connections, and show that the networks which you've produced using the exponential distance rule actually very, very faithfully mimics the efficiency changes that you can observe as you remove connections. So the point I want to make is that this distance rule, the exponential distance rule, actually gives you a handle on looking at global properties, not just not just the motives and the click, which I was talking about earlier on. And the click actually we're coming back to the core of the previous speaker. So that's, it's not without its own significance. But there is, the strength of connection does give you a very, very strong degree of specificity. But again, it requires looking at the strength of connections. And I think one of the reasons why this has been perhaps ignored in the literature is, first of all, you can't see this kind of range of strength of connections with diffusion MRI. You can see it with track tracing. If you're going to do this with track tracing, then you've got to count neurons or synapses, and that's a lot of work. So if you want to have that kind of data, then you can't just do what a lot of us have been doing for many, many years, myself included, which is saying strong, weak, and medium. Strong, weak, and medium isn't going to tell you about the range of strengths of connections where the specificity is coming in. It requires a much more thorough approach than that. And what that shows you is that you have these 5 orders of magnitude. That means that the counts that you're making run into very, very high numbers. And that's an important point.
But the other thing that I would like to understand better then is, so if I take now these scaling laws that we have for weights and for the probability to connect, right? These are probabilistic. They're probability distributions. So I just take a lattice of units, and I'm going to wire them up following these 2 probability distributions of lateral connectivity and the strength of these connections, right? Then you would expect, if I start to sort of chip away those guys and I remove the weights with the smallest, the lowest value, the lowest strength, that I would get also the random patterns. Because these 2 scaling laws or wiring laws themselves don't give me any kind of symmetry breaking, right? So if I do this 1,000,000 times, I would have 1,000,000 different kinds of topologies, or not?
No, you don't. What you find when you take the data, the inter-aerial connectivity database that we have, which is 29 areas and some hundreds of connections, and you remove the weakest connections, you get a backbone. You find the backbone of the cortex, and that gives you there's features and characteristics of that backbone. When you do the same thing to your random networks that you generated using the exponential distance rule that we have, what we're saying that is a probability. So you pick more frequently the very strong short distance connections, what have you. when you remove the weak connections and you end up eventually with your backbone, you find that it has many of the features of the backbone in the data that you've observed.
Which features do you recover best?
Well, I mentioned the motives.
Mm-hmm.
That's pretty good. The correspondence between the motives is actually excellent.
Mm-hmm.
So you have 16 different motives of, say, free nodes. Of those 16 different motives, your distribution, which is captured by the exponential distance rule, is actually excellent.
Mm-hmm.
Then I referred to this question of hubs. Now, the people have been finding hubs, as to say, areas with high degree distribution to them, a large number of connections with other cortical areas. And the idea of the cortical core is that the hubs form more connections between themselves than you would predict statistically. So that's actually what is referred to as a rich club. The rich club analysis was introduced in network science by Coriza and other people about 7 or 8 years ago. Now, you can't do that kind of analysis on a high-density network. The normalization doesn't work. So what we've done is look at the cliques. Mm-hmm. So the cliques are sets of areas which are 100% connected amongst themselves. And we find a very, very large number of cliques. And when you look at the probability of finding that by chance, it's extremely low. When we do this same analysis of networks which we've constructed using this EDR, we find the same number of cliques with a very, very similar frequency. And the correspondence is really, truly very remarkable.
So, and you're not imposing any other constraints. You just follow this exponential distance rule, and that's it.
Absolutely.
Yeah. Okay.
So some fairly simple geometric rules are giving us a lot of information about connectivity within the brain. Do you then draw inferences about the developmental processes that are building brains? And because this has been a week where we looked at evolution and development and their impact on the way the adult brain is. And your data, I guess, gives hope to people that want there to be some useful developmental rules that we can apply, perhaps to build a brain on which experience then has some impact.
Right. I think that's a very important issue, which we haven't looked at, despite the fact that I have another side to myself, which is to do with cortical development. So I mentioned in my presentation that there's this log normal distribution of weights. A number of studies have looked at the distribution of synaptic weights at the single cell level and also find a log normal distribution. And there's a recent paper review in Nature Neuroscience looking at log normal distributions in frequencies of firing and other sort of phenomena. And it seems to be a sort of signature you're finding a lot. Now, the log normal distribution in itself suggests that there could be a very simple algorithm which would be controlling outgrowth of axons. Now, for many years, I was interested in the formation of connections, and that was in cat and monkey cortex. And in those days, the predominant theme in cortical development was this notion of exuberance, that you had an overproduction of connections, and that the selective pattern, the precise pattern which is characteristic of the adult, emerges through a pruning process. And that this pruning process is actually necessary because there's not enough information in the genome to set up the correct connectivity. We challenged that, and every time we challenged it, it turned out not to be the case.
So what did you find?
Well, in the case of interhemispheric connections, so that was the model which was hugely used between 1970 and 1985. By Giorgio Innocenti, Doug Frost, and many, many people were looking at Herb Kalaki. There you could observe a widespread connectivity in the very young animal, which would decrease as the animal would mature. So we challenged that by looking at the callosal connectivity of the prenatal monkey in the visual system, and the characteristic of the monkey is that the area V1 is acallosal in the adult. So then you can really ask the question, is is it acallosal during development? Because it shouldn't be if this exuberancy rule is going to be general, and it's not.
Mm-hmm.
We were able to show that there's never a causal connection going into area V1 of the monkey. So I think it's very much a problem. You've got to be able to distinguish with the growth of the brain and the expansion and the increasing and therefore the decreasing density simply because of the expansion. You've got to be able to sort that out from the actual creation of the specificity. So that was into hemispheric connections, but we then did the same thing looking at the connections between V2 and V4, which are you have these bands where you have projections to V4 and projections to MT. And we were able to show that they're correctly aligned from the word go. There's there's an overall decrease in density, but it's not the decrease in density that creates the pattern.
Mm-hmm. But then this old-fashioned idea, we can call it now, of exuberance and pruning would still hold, but for a smaller set of let's say, neurons and connections? Or you think it's really the wrong way to think about how the system gets wired up?
I think it might be an oversimplification. I think the idea that you don't have enough genes to specify connections is not really very interesting. I think the confusion which was made was not, is there a decrease in density of connections with growth? There is. The question which was posed was, is it that decrease in connections which creates the specific pattern? And that, I think, has never really been satisfactorily shown to be the case. I think it's not, I'm not saying it plays absolutely no role at all, but I think there's much more guided growth and target specification.
But couldn't you argue that if you apply the exponential distance rule to a developing brain, you should see something that might look like pruning by definition, because you're imposing a distance relationship of your connectivity in an expanding volume?
Well, I think that would be an interesting thing to look at. Simply look at the brain and see how does this exponential distance rule look in a very immature animal. The problem is that the numbers get very big, and you really need much more automated techniques in counting, but which are coming through actually.
Right.
So I think that sort of thing is something that will be addressable.
Mm-hmm.
So one of the other things that you discussed today was how the primate brain seems to minimize the wiring length as a consequence of applying these rules. But bearing in mind this point about changing brain size, you also discussed some data about animals with smaller brains like mice, and that the same rules don't necessarily apply. So are there some scaling issues here for brains that perhaps would help us unravel some of these issues?
Absolutely, yes. This is something we're very interested in looking at. We've with Zoltan Torokai and his postdoc, who's now back in Romania, we have funding with John Cass to look exactly at that. So we're looking at the mouse and the microcebus, which is a small primate, and we want to look at the baboon, which is about as big as we can go in primates. That's not a huge deal. And the idea is to see how this exponential distance rule plays out in changes in brain size. So we were still working with our own database, which we're putting together with Andres Burkhalter. But to keep us sort of going on this, we've been looking at the Allen Brain Institute database, and it's actually really rather interesting. So this is very, very preliminary, and with Zoltan Torokai and Ken Koblock and our colleagues, we're still analyzing this data. But the glaring thing is in the mouse, there's a huge amount of wire economy that you can impose on it. So the areas in the brain are not localized optimally as they are in the primate. And that makes it look straight away very strange, because you have areas which are not connected, which are actually quite near to another area, and they're not connected to it. And that's what you absolutely don't see in the monkey. And the thing that we're very interested with Torokai and the group in general is to see how folding comes into this. So the comparison between mouse and monkey is a bit complicated because we have a change in brain size, but we also have a change in folding. So what we've been doing is, with David Van Essen, is we've been, instead of taking the distances which we've been using up until now, which have been white matter distances between areas, approximating the trajectory of the axon, we've been taking surface distances. And when you do that, you're sort of unfolding the brain, as it were.
Right.
And when you do that, you get a very, very different distribution of distances in the monkey. So this is unfolding the monkey brain.
This is flattening the cortical sheet, in effect.
Virtually, yeah. And then when you look at your distance distribution, you get something much flatter. It's not at all like a Gaussian, a very pointed Gaussian. And it looks just like the mouse.
Mm-hmm.
It looks just like the mouse. And so we're now playing with that and seeing how this distance distribution interacts with this exponential distance rule to set up the specificity that we're seeing.
So what kind of rule would hold in the mouse brain?
That's a key question. Certainly the exponential distance rule is part of it. But the jury is out completely. I mean, Zoltan Tarakai and Maria Havaz is doing simulations on that, and we don't quite know.
Mm-hmm. Mm-hmm. But you seem to suggest this morning that the probability to find long-range connections is higher in the mouse brain than in the macaque brain, or did I misunderstand that?
No, that's correct. So when you look in the macaque, you have an exponential lambda value with a very sharp drop. When you look in the mouse, the decline in strength with distance is much more shallower. Mm-hmm. And that's certainly part of the exponential distance rule being altogether specifying much less specificity in your mouse. So we talked about the cortical core. In the macaque, you have this very large number of clicks, which is extremely improbable. In the mouse, you have a much smaller number. Mm-hmm. And don't forget, although the overall number of areas are the same in both species, we're actually making these networks on the same number of areas. We're using the same comparable number of areas. So the cortical core is much less defined. So what I think this is pointing to is in a world where one might be tempted to say, well, the mouse can be a very interesting model for the brain. It might be because you can have knockouts, you can have knock-in, you can use optogenetics very easily and what have you. But simply from these kind of large-scale properties, it appears to be very, very, very different.
Mm-hmm. But in the areas you're looking at, the cell densities are comparable to primate?
No, they're not. So the scaling rules, and John Kaus could talk about this. He's with his colleague Herculano Huzel. They've done a lot of work on that, and the scaling rules in primates and rodents are quite different. Mm-hmm. So basically, I mean, the way I've understood it, in fact, I want to talk to this about John. This meeting is that as the brain changes in size, you can change the density of cells in rodents. My feeling is that this might lead to a capacity for miniaturization. The microcebus is about a centimeter and something, and it's the smallest primate brain that exists. And as the lady this morning was pointing out, there's a huge radiation in primates. There's a huge adaptation. The mouse, the rodent brain can go down to tiny little things. Mm-hmm. I mean, they have moles and voles and what have you that have...
No, but this means, Henry, that possibly the scaling law still holds, but it is modulated by cell density. It scales in turn with cell density, right? So the cells are packed more tightly, and that gives you an exponential decay. What if the cells will be packed more loosely? It gets stretched out. Would that be reasonable? I think that's
what Herculano Huzel's results are saying. I think she's saying that the density has a much bigger variation in rodents, so that you can make smaller and smaller brains by making smaller and smaller cells.
But the other thing about the mouse example that might be problematic is that for the primate brain, you were saying, look, these laws we have on connectivity tells you something about the optimal wiring of a brain, because you see that regions that are connected are placed together and so on, right? And you make this distinction, or the example of the ventral dorsal visual stream. However, now for the mouse case, you say that you find regions there that are adjacent but not connected.
Not adjacent, but nearby, yes.
No, but okay, still, right? So they're nearby but not connected. So that seems to be a violation of the principle that you identify. So that means a mouse brain in its development is really setting up fixed borders. So no, we're not going to wire these guys up.
Yes.
While you apparently are not doing that to the primate brain. Is that correct?
Exactly, yes. That's what I found so very, very surprising. I didn't expect that at all. Yeah. And if you optimize your mouse brain, so you place the areas optimally so that you don't have these absent connections, as it were, and then you look at your lambda value, it looks much more like a monkey. So you can convert it into a monkey. Now, how we got to that state, I don't know, but I'm wondering if the ancestral primate was actually, I think, probably quite large, and so today's rodents might have undergone a miniaturization. And is that the cost that you're paying for miniaturization, or is it another kind of adaptation that we're not putting our finger on?
So about optimality, what I would like to understand is how you define that. Because if you talk about optimally wiring a brain, what does that really mean? Does it mean like that you have a constant number of wires crossing certain distances independent of where you are on this cortical sheet? Does it mean that you want to optimally transfer signals between areas, that you want to use as a minimum number of intermediate steps to get from A to B? What's optimality here?
Well, I've been using the term maybe rather loosely. What we're talking about is optimal placement. Can you replace the areas in a monkey brain in such a way that you would be using less wire given the strength values that we observe? And the answer to that is no, you can't. There's no way you can do that. These are simulations that take days and days and days to do, but there's no way of doing that either to the binary, or maybe there's one or two areas that you can flip positions, but the weighted absolutely not. In the mouse, you can get 15% reduction in wire both for the binary and for the weighted network. So we're talking about optimal placements. The second part of your question touched on this thing that I was talking about, that if you take this exponential distance rule to heart, and you look at the visual cortex, for example, and you look at the central visual one and peripheral area visual one, you'll notice that they're in very different neighborhoods in terms of the areas which are surrounding them. That would suggest that the central area V1 should be more strongly connected with a very different population of areas than the peripheral V1, and the same for V2. And we've looked at that, and the answer is that yes, the connectivity is very different. So central V1 is in front of the temporal areas. So you find that central V1 and central V2 look like ventral stream areas. And if you look at the peripheral representation, they're in front of the parietal cortex, and you look at the strength of the connection and you do a summation of strength, then these areas, or these parts of these areas, appear to be dorsal stream areas and quite different. So from that, I'm wondering if we have an optimization of the position of areas, but we also have an optimization of the shape of areas. And if you look at the flat maps of David Van Essen and others, they have very, very distinctive shapes. I mean, you remember the motor cortex and somatosensory cortex, these long, thin strips of cortex where you put the whole homunculus in this peculiar sort of strip. You could imagine something very different.
But would you accept the hypothesis that what you're optimizing is a transduction delay between these areas?
Well, I mean, I think that was part of the motivation that we had to look at the distance through the white matter. So we felt that we were looking at and I think in the first instance, we measured the white matter distances and the surface distance, and you could imagine that these distance relationships we've been reporting could simply be reflecting change of the cortical property, in which case the surface distance would be very good. Or it's something to do with transduction of signals, in which case your trajectory through the white matter should be very good. And so I was expecting to have a sort of black and white answer comparing those two, and that didn't happen.
What did happen?
Well, not much, actually. We stuck to the white matter distances because that's what we had started off with. But then when we were making this monkey-mouse comparison, that's where we asked ourselves the question, what about if we unfold the monkey cortex and how will that behave? And we're still looking at that, but it certainly changes the distribution of distances.
But so when you say that you want to do this minimal wiring test and see if you can rearrange cortical areas to reduce the wiring, how do you deal with the fact that the pieces of the jigsaw are all very different shapes and there's really only one way it fits together? So if you rearrange it, how do you compensate for that?
Oh, well, so what we're rearranging, we're not tackling shape here.
Right, okay.
Tony. No.
Yeah, and I presume you're not.
Yeah, so we're changing we're measuring distances between the barycenters of the areas. And so we're switching barycenters around in space. We're not trying to shift these peculiar shapes and make them all fit in. It's not a jigsaw puzzle thing. So we're moving the barycenters, and we're considering the distance from one barycenter to another is the distance between one cortical area and another.
But as you say, the shape of these areas can be quite extreme to long, thin strips.
Yeah,
which then does have an impact.
That introduces its own problem about what is a barycenter of a long oblong shape. Yeah, but that's where we are.
So then, if you had to choose one objective that these wires are optimizing, it would be just to minimize the physical wires between areas. That would be your bet today.
As I say, I'm not sure. If you look at area V1 and V2 in the macaque, it's the two biggest areas in the macaque brain. In fact, they're very, very large. If you, relatively speaking, compared to any brain, they're huge. And in the central representation, they're folded in such a way that V1 and V2 lie opposite each other, separated by 1 millimeter of white matter. So it's the most extraordinary engineering to make sure that you've really minimized the distance between the two biggest areas of the macaque brain. That tends to make one feel either it's because, if you had as much white matter in your brain as a mouse, it would be the size of a bathtub, which would make getting out of the door rather awkward. So we know that there's a reduction in white matter, there's a reduction in total connectivity as brains get bigger. So it could be that piece of engineering is dealing with that problem. Alternatively, it's dealing with the transduction problem, and you really need that kind of very, very short distance for V1 and V2 to do its job.
And so, in fact, that could maybe explain some of these scaling distances, is that the amount of wire you have in the brain really becomes under pressure as you get bigger brains. And so you really have to optimize for wire length in a way that in a small brain is not so important.
Absolutely, yes.
But now another element of this is that we could argue, well, you measure this in adult monkeys, right? So this is a cortex that has been learning and changing its properties due to plasticity rules. Plasticity rules are activity dependent. So what you're looking at is really the history of activity in this system. Now, the history of its activity is strongly constrained by subcortical systems. Like in the case of cortex, you'll depend on your thalamus. And now, thalamic projections are not random. Thalamic projections actually also define very specific envelopes of interaction with cortex.
Absolutely.
So I could argue what you're actually measuring is an echo of the combination of the envelope of thalamic projections into this system modulated by the local plasticity rules that make these guys wire up together. Would you accept that interpretation?
I'd go much further than that.
Okay.
The work we've been doing with Colette de Hay over the last 10 years shows that the thalamic fibers release a mitogenic factor, which governs the proliferation in the germinal zones. So the size of your areas, the size of the cortex, is very largely determined by the interaction between the thalamic fibers and these germinal zones. And that whole relationship between the thalamus and the cortex setting up, for many years, people thought that the thalamus was playing an important role in the specification post-mitotic. So you had the barrel cortex and the effect of plucking whiskers and seeing the representation of the barrel's changing in some fashion on the cortical surface. So what we've been arguing for a long time now is that the thalamic fibers are actually their primary target, and particularly in primates, it's much more accentuated, is not the cortical plate, it's the germinal zone.
Mm-hmm.
So the thalamic fibers get into the cortex in the primate very, very early, before there is any cortical plate. In fact, before there is any release of mitotic neurons into the cortex. And what they're doing is actually, I think, hugely to do with shaping the proliferation and possibly the specification.
Would it also mean that if we look at these two types of thalamic projections, like parvo-magnocellular, right, where the parvocellular seems to be not very plastic and the magnocellular is, shouldn't there then be a correlation between parvocellular projections and your scaling law?
Well, for me, the scaling law is a little different from that. It's to do with how white matter, grey matter changes over a range of brain sizes.
Mm-hmm.
And so what people have been able to show is as brains get bigger, the volume of white matter doesn't increase at the same rate as the volume of grey matter. And so this is where this notion that was introduced by Ringo some 20 years ago, that as brains get bigger, there's a huge pressure to economize numbers of connections, which if you think about it is really rather extraordinary, because brains are all to do with connections, and you've got to actually reduce the number of connections and reduce the number of long distance connections. So that for me is...
but the question is, how do you do that, right? How do you control the developmental program to achieve this, that you economize on these connections? So then the question is the thalamus then the key to understand the genesis of your scaling law?
Well, I don't know. I think if I understand Huzel's work correctly, as your rodent brains get bigger, the size of the neurons get larger. So you have this change in density, which you don't have in primates. So that seems to me to be a very different sort of algorithm for building the brain. If you say, well, okay, we're going to have primates, and primates are going to have basically a small variation of cell size, and we want to go from a small brain to a big brain, so we're going to economize on connections. And then you do the same thing for rodents. You say, well, we're going to have to do the same economy on connections we did. That's a kind of given. But here we can change the brain size, the neuron size a bit more. So you seem to run into a bit of a problem with creating very big brains. You have you have these South American capybaras, you have these big South American rodents. But then I'm wondering if the adaptation is much more adapted to making small brains. So that is the sort of rules I see for the scaling. I haven't been thinking about how the
because what I'm after, what I try to figure out is, what we're trying to do this week in the school is this relationship between genetics and development, right? And as we discussed earlier, what you measure with the scaling law is like an echo of these two processes working together.
Right.
So then I was trying to push you a bit on trying to come to some understanding, okay, what are the causal factors that give rise to these scaling laws that you measured? Right.
Okay, well, we're going to look at a particular case which would probably come back to that. You have this situation of microcephaly, where you have very, very small brains. And there's a lot of interest in understanding that, because expansion of the brain is very much a hallmark of human evolution. And so microcephaly raises something of a question about our evolutionary origins. People with that condition actually show remarkable levels of cognitive capacity. And so we're now going to start working with a mouse, which is a model for this. And so it will explore what are the genetics regulating brain size and how that will touch on this exponential distance rule, and also on the whole relationship between brain size and the exponential distance rule. So these are questions which you will address in the future. Address in mouse, yeah.
Okay. Tony, you have any more? Okay, so Henry, now looking at this, your tour through, let's say, the anatomy of the brain that's going on for quite a while, and which also you have made amazing progress. If we want to follow in that trajectory, what's Henry's law that we should adhere to?
I think there's a number of cases, without saying any names, where people... do you remember the decade of the brain?
Mm-hmm.
Right.
Very well.
So the decade of the brain was spurred by a book, I won't say the name of the book, which said, well, we've got people endlessly producing data, and you go to the SFN meeting and it's so depressing because you've got miles and miles of posters of people showing endless data. What we now need is somebody to come along and give us a model of this, much in the way that Watson and Crick were able to do with the double helix. And we'll have a kind of eureka moment, and we'll suddenly understand everything, and we will have understood the brain. Basically, we would have done it, gone there. It would be a finished story. We can pass on to something else. I find this absolutely ludicrous because it's based on an idea, and there's at present a European project to understand the brain, and it has this idea built into it that we've got a lot of data and we can go back and accumulate over 150 years of journal of comparative neurology and skim through and take out all this data and pile it up together, and it will add up to some total explanation. And I think this is dangerous. I think it underestimates the challenge to understand intelligence, biological intelligence. I think it underestimates the challenge of relating that to neurological principles. It forgets the fact that any experimental result is done in a certain intellectual framework, and the interpretation of those results is not extendable. You can't extract this information willy-nilly and apply it across the board. So I think that either governments are going to decide that understanding how brains work is worthwhile, and they will provide money to do basic research, which means not endless science papers and nature papers and what have you, but actually pay people to how many synapses do you have on your pyramidal cell? What is the connectivity of your average whatever, and pay people to do that kind of work. Mm-hmm. And so I think that there's a need for empirical data. I think that the power of simulation is fantastic and has to go along hand in hand. But if you don't actually have the investigations, if everything's got to be a breakthrough now and then, immediately, if you're only going to have high profile kind of projects, then you're going to find that you're actually playing around with inadequate data. And I think that this small world thing, it's not that there isn't a small world complex network. There is. The point I want to make is it doesn't exist at the inter-aerial level. And there's been over, I don't know how many dozens of publications claiming that is the case. And they've been using inappropriate data. Somebody's got custard on their face. Mm-hmm. And I think that the willingness to challenge the big questions, I think there needs to be much more interaction between people doing the experimental work and the people doing the simulation. And I think these have got to be hand in hand. And this is exactly what we've been trying to do with Zoltan Torokai and now with Xiaojing Wang and others. And I think it's tremendously exciting because you can see your anatomy in a much larger framework, in a much bigger context. And I think this will ultimately lead to real breakthroughs. Mm-hmm. I think collapsing things and overselling and saying, well, we're going to have a decade of the brain and okay, we need big science. That's for certain. But we also need reasoned science, and we need empirical data.
Mm-hmm. And now, 4 years from now, Tony's going to visit you in Lyon.
Yes.
Have some pâté with you.
No, Andouillette.
Okay, even
mm-hmm.
better.
Mm-hmm.
But he's also going to confront you with the prediction you're going to make today. So what's the one specific prediction you would share with us today that Tony's going to check out 4 years from now?
Well, I think that we're going to find that there's a very large range of solutions that biology has brought to the brain. The dream that you can have one brain and extrapolate from that and understand the brain principle goes right against all our understanding of zoology and how it works. So I think that when Tony comes to Lyon, we have a pot of wine, white wine, and an andouillette. Andouillette, you have to have very, very white wine, very, very dry white wine, and in very large quantities. Is that we'll know something about different sizes of brains and how folding interacts with these things. And I think that the idea that you can use the mouse brain as a model brain for all brains will be seen to be completely fallacious. I hope between now and then somebody else would have done the local circuitry, because this is where the machine really is. This is where the machine lies across different brain regions. At the moment, we have no idea about the local circuitry of area 46. Mm. We know the local circuitry of V1 in the cat, and we're extrapolating that across all species, all brains, and what have you. So I think that we'll have a much more deeper understanding of the variability of brains and the solutions they bring.
Great. Henry Kennedy, thank you very much for this conversation.
Thank you very much.
I hope it was not too boring for you.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.